Hello everyone, my name is Ashish Katyal. Today we will learn about a new topic, that is Genbank. Genbank is a common terminology that is used in NCBI. Sometimes Genbank is also known as database, while sometimes Genbank is also known as format. In today's experiment or in today's session, we will learn about Genbank format. Genbank is commonly available in a website that is known as NCBI, or it's a database known as NCBI, National Center for Biotechnology Information. Before proceeding towards this topic, we should first learn what is database. For example, if I'm saying a database is a term that is commonly used in bioinformatics where we can store certain amount of data. For example, I am adding certain kind of DNA, RNA or protein data into a place, and at the same time I can also retrieve that data. Then this kind of thing or this kind of place is known as database. There are two kind of database that are available in bioinformatics. One is known as sequence database, another is known as structure database. For example, in case of sequence database, most of the things are related to RNA, DNA and protein sequences. While in case of structure database, most of the time the structure data is of proteins. Other than that, the DNA structure database are also available, but most of the time we are dealing with protein database. Along with that, if I'm further classifying this sequence database, there are two kind of sequence database that are available. One is known as primary sequence database, and another is known as secondary sequence database. As the name indicate, primary sequence database is also known as raw database, in which all kind of experimental results are available. For example, I am doing certain kind of work in my lab and I extracted certain kind of DNA or maybe protein sequence, and I want to submit that sequence into that particular database. So it may be correct, it may be incorrect. So it may be relevant or it may be irrelevant. So this kind of information or raw information can be stored under primary sequence database. There are three common primary or fundamental sequence database that are available. One is known as NCBI, another is known as EMBL, and third one is known as DDBJ. As the name indicate, NCBI, as I earlier told you, National Center for Biotechnology Information. Second is known as European Molecular Biology Lab, and third is known as DDBJ, that is DNA Data Bank of Japan. In case of secondary sequence database, the most common one is TREMBL and second one is UniProt KB. TREMBL means Translated European Molecular Biology Lab, while UniProt means Unique Protein Knowledge Base. Other than that, if I am focusing on structured database, there are two kind of structured database that are available. Similarly, as in case of sequence, there is primary, secondary. In case of structure also, there are primary and secondary. For example, in case of primary structure database, one is known as CATH, another is known as SCOPE. CATH means class, architecture, topology, homology, while SCOPE is known as structure classification of proteins. The secondary structure database common examples are PDB, another is known as MMDB. PDB means protein database, while MMDB means molecular modeling database. If I want to distinguish between primary and secondary database, I can say that primary database consists of relevant and irrelevant things, while if I only filter relevant things out of that and I'm storing somewhere else, then it becomes secondary sequence database. If I am filtering again secondary sequence database and storing somewhere else, for example, I am classifying that whether that particular gene sequence consists of certain kind of motifs in terms of protein, then it becomes primary structure database. And if I'm again filtering that thing and becomes secondary structure database, which is the purest form, that is 99.9% results are quite correct in case of PDB as well as MMDB. So we are currently focusing on primary sequence database, specifically NCBI, EMBL and DDBJ. The other name of primary sequence database is fundamental database. As we can see in the further slide, the name indicates primary and fundamental database. As I earlier told you, database consists of either storing something or retrieving something. So in case of bioinformatics, there are three different kind of database that are available. Primary database that are available. One is known as NCBI, as the name indicate, National Center for Biotechnology Information. It is under which Genbank is there, where we store all kind of data. Genbank is format also, but right now I am talking about the database. It is maintained by NCBI and it is available at US. Another is known as EMBL, or for specifically for nucleotide, it is known as ENA, that is European Nucleotide Archive. It is maintained by EMBL or EBI. EBI is known as European Bioinformatics Institute, while EMBL is known as European Molecular Biology Lab, and it is maintained at United Kingdom. Third is known as DDBJ, that is DNA Data Bank of Japan, and it consists of all kind of data which is related to nucleotide or maybe protein or maybe any other thing. Specifically, today we are talking about the database that consists of the primary information. So it is maintained by GenomeNet and it is available at Kyoto in Japan. So these all database exchange their information on regular basis. In 1986, first time EMBL as well as NCBI has collaborated, and after an year, DDBJ also collaborated with NCBI and ENA, that is European Nucleotide Archive, and they make a repository known as International Nucleotide Space Database Collaboration Repository. According to this organization, they can exchange their data on the daily basis. Also, these are the websites that are available for by which we can see the web page or you can say the home page of this particular database. For example, NCBI, EMBL, and DDBJ. One thing also I want to highlight is that, for example, in case of a browser, for example, Chrome is a browser. The search engine is Google, or maybe there is other search engine that are also available like Rediff or maybe Bing or maybe any other search engine that are available. Similarly, there are other kind of browsers that are available such as Chrome, Mozilla, maybe you can also say that Safari is also there. So similarly, if I'm considering this NCBI or EMBL or ENA or DDBJ as a browser, so there are specific search engines that are also available with them. For example, in case of NCBI, the search engine is Entrez, ENTREZ. In case of ENA or maybe EMBL, the search engine is SRS, that is Sequence Retrieval System. While in case of DDBJ, the search engine is DBGet, that is database get. So these are the three different kind of search engines that are available with these browsers. Along with that, I want to first highlight this thing which I've already told you, that is these search engines are quite helpful in searching our query. For example, I want to search a sequence of insulin specifically related to Homo sapiens. So I will go to this NCBI and I will type insulin and human if I specifically want that particular refined research. Who will perform this search? This particular thing, that is Entrez. So we will see this thing while we do hands-on session. So the first thing which I have told you in case of database is submission of data. If I want to submit small kind of data, then we use Bankit, and if we have to submit large amount of data, then we will use SequIn, sequence input. So these are the two kind of data submission which we will use in case of bioinformatics. Another thing I want to say that this is the screenshot of SequIn in which we can start our submission, or we can read our existing record which is already available in our database. So you can also see database submission here. The format is Genbank format, or we can also use EMBL format. Before going towards this format, we will see how we can store the data. So I am not going into the detail of this thing, but I am showing these how many types of database are there and how can we store our data, and which are the tool which we can use to store our data. As you can see, there are huge amount of data that is coming from year after year, and after COVID we have seen a spike of data that has been coming day after day. You can see genome, there are lots of genome that are available. By using this link, we can see how many organisms has been sequenced. In this case, you can see this much number of eukaryotes, this much number of prokaryotes, and this much number of plasmid and organelles genome are already sequenced. So you can see there are lots of entries that are available. Other than that, if I want to focus on for example this particular data, means how many genome or whole genome sequencing has been done, so you can click on this particular thing and you can see this is the kind of thing. First it has, you can see this is the same graph which I have shown in our slides, and after that you can see this is the number. For example, it has been started in 1982, and after that this particular thing has been increased in such a way that in 2000, for example, right now it is 23, So till December 2022, this much amount of data has been submitted in NCBI. So there is a huge exponential amount of data that are available in our database. To churn that information or to find out information out of that particular primary database, we need certain kind of bioinformatics tool. Before going toward this particular tool, this is the same screenshot which I have shown you in the previous web page, in which October 22 record is available till here, but in that December 22 results has been shown. So these are the exponential amount of data in terms of genome as well as whole genome sequencing has been done. So you can see date, base, sequences, all the things that are available. So before going towards this second thing, that is retrieval of things, we need a common language so that we can communicate with the database that this much amount of information I need in this particular format. So we need format. For example, if in a college I want permission, then I will write to my teacher that in a form of application. Application is a kind of format. So I need a permission, I will write in the form of application. While if I have to convey my information to whole college, then I will write a notice. So these are the two different formats. Similarly, in case of bioinformatics, various formats are used. As you can see, FASTA is most common format that is used, which is simplest in nature and very fast, because it only consists of minimal amount of information along with maximum, or you can say most of the sequence information. Along with that, the second is known as Genbank, which today we will currently discuss, which is fully informative in nature, and it consists of lots of data, or you can say data about data or metadata. Along with that, the sequence information. So if you want detailed information, then you will go for the Genbank. If you want minimal information regarding the sequence only, or other optional information are also there, then you will go for FASTA. Other than that, there is EMBL format for EMBL database. Another one is PIR, that is protein information resource. Other is GCG. Another is ISF, IntelliGenetics sequence format. Other is multiple sequence format. These are the most common format that are used in bioinformatics. Today, our experiment, or you can say our discussion, is based upon these two formats, that is known as FASTA as well as Genbank. But we will see the type of format or how they look in bioinformatics, these different formats. So I'm going, for example, FASTA format. The main identification of this format is greater than sign. If greater than sign is available, then we can say that it's a FASTA format. Along with that, it consists of accession number. Accession number is a unique number, or you can say as in case of bank, there is a unique ID, 16-digit ID is there. So similarly, in this case, alphanumeric ID is there, which is known as accession number, which is related to that sequence only. Along with that, there is a description about that organism, the type of sequence that are available. These are all optional information that are available. For example, peptide is there, complete coding sequence, CDS means coding sequence is there, and the length of the sequence is also given. Along with that, the major thing which is available in FASTA format is sequence only. So this is the DNA sequence that is available. So you can say that in case of FASTA, the minimum information is that is required to write this format is greater than sign. For example, I am writing my name, Ashish, and after that I want to write the sequence. For example, I am writing A, T, C, 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 T, 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 G, G, G. This is a kind of FASTA format. I do not need this information for this particular thing. So in case of FASTA format, it is quite simple and it is quite fast for performing any analysis. Second is known as Genbank format, which is also known as the most informative format, in which lots of information are there. For example, locus number is there, accession number is there. For example, there the size of the sequence, the type of the sequence, either it is linear or it is circular, the type of submission, the detailed definition of that particular thing, the sequence information, the feature information, all the things are available along with a identifier, that is // So this kind of in case of FASTA, it was greater than sign. In case of Genbank, it is // which is the identification of Genbank. But there are other formats that also uses this // // For example, EMBL format also uses this. As you can see, in this case of EMBL format, certain things has been changed. For example, here it is ID rather than locus, and this is the same thing which has been given. Again, RNA and primates, the base pairs, the accession number. You can see in case of the previous one, accession number is written in full. Here it is written. While in case of EMBL, the accession number is written as in the form of certain abbreviation, that is AC. For example, definition is written in the form of DE. Same thing are available, and sequence is available in the form of SQ. So various abbreviations are used. One thing you have to keep in mind while looking at this EMBL format as well as Genbank format, in a single line only 60 base pairs are there, as you can see, which is further fragmented into 6 different fragments. Each fragment consists of 10 base pair. This format remains same in case of EMBL also. So you can see here also, one it is starting from here and 60, next is 61, other is 120, and it has been further fragmented into 6 fragment, where each fragment consists of 10 base pair. All the things are available. So you can see here 360 is there. So 361, 2, 3, 4, 5, 6, 7, 8. So total is 368. All the things are available, and it is quite well formatted. Other format is NBRF format. So, or it is also known as PIR format. It has also same recognition as that of FASTA format. So you can see here greater than sign is there along with that, but there is P1 information. For example, P1 is related to proteins. It is specifically used for protein, as the name indicate, protein information resource. Along with that, so there is certain other symbols that are also available. So you can see here the information is related to protein sequence only. Other than that, there is GCG format, which is specifically used for DNA, and it tells about the ratio of ATGC that are available. And you can see in the previous EMBL and NCBI, in a single line 60 base pairs are used, but in this case 50 are used. So there are specific points that you have to keep looking while seeing this particular kind of format. Other than that, there is IG format, IntelliGenetics format, in which it starts with a semicolon, and after that certain information is given in terms of, you can say, sequence, length, or maybe date of submission. All the things are available. So other than that, multiple sequence formats are there. So you can see if I am taking multiple FASTA file, for example, one is of human, one is of rat. So for example, human I am writing here, while other is rat, another is chimp. So these 3 different kind of sequence are there. I am taking, if I am taking 2 or more than 2, then it becomes multi sequence format. But today's experiment is focused on both, that is FASTA as well as Genbank format. So other than that is PDB format. It's a structural file format in which all the coordinates are available. X, Y, and Z coordinates are available. It is related to proteins. As the name indicates, it indicates atomic coordinates, and along with that it starts from N terminal and it goes towards C terminal, because in case of amino acid, you can see there is R group, there is H, there is COOH, and naming is going from N terminal to C terminal. So this is the thing. Along with that, the various type of format, we have to focus on Genbank as well as FASTA. So first one is FASTA, second one is Genbank, third one is EMBL, fourth one is PIR, other one is GCG, other one is ISF, and other one is multiple sequence format. So as I earlier told you, these 3 database met together in 1987 when DDBJ joined. Then they said there should be a common feature table format that should be available, that gives a complete detailed information about that particular sequence, and they agreed that there should be a common format which should be in a file format, tabular format. For example, Genbank format, which is also known as Genbank flat file format. Flat file format means it gives lots of detail from the sequence related to that particular organism, or the authors which are available, which are associated with that particular discovery, or that particular discovery, or maybe invention of that particular theory. Sorry. Along with that, they have a common standard for annotation practice. If someone change, for example, as I earlier told you, here it is definition. In this case, it is definition, D is DF. For example, here it is accession number, here it is AC. All the things are common, remain common in this particular database, that is NCBI, EMBL, as well as DDBJ. So in this case, we can see. So we can see that in this case, various kind of common format is used. For example, here we are talking about Genbank. and Fasta. So Fasta, as I earlier told you, in this case, greater than sign is used. If greater than sign is available, then we can say that it's a Fasta format. Along with that, some optional information is there. Sometime at the end, asterisk mark is also there for the reference sequence, and sometimes accession number is there. Along with that, name of organism is there, the type of organism, or you can say the type of the sequence. For example, here it is showing complete coding sequence along with the length, which I have already told you in the previous slide. So along with that, there is a sequence information that is available. In case of GenBank format, there are three different things that are available. One is known as header, another is known as feature, and third one is known as sequence. One other thing or one other identification of GenBank format is double forward sign, which I have already told you in the previous slides. So in this case, you can see a detailed information is given. Either it is related to locus number, either it is related to sequence, either it is related to the type of sequence. For example, mRNA is there, whether it is linear or circular, whether it's a primate or any other organism, type of organism, or here it is the date of submission. The definition is there, accession number is there, origin is also there. So origin means where it is started. For example, here it is starting and here it is ending. So the length it is 368, which I have already told you, but here it is already written. So all the kind of information you can get here. Feature thing is missing. So this is known as header. Along with that feature in which all the detail information is there and sequence information is there. One thing I again want to mention is that in a single line, 60 base pairs are there in case of GenBank format, and each line is again fragmented into 6 parts, where each part consists of 10 base pair. We will see in detail how it looks in a real sense. So one is known as header. As you can see here, you can see there is an accession number which is there. So here is the accession number. One thing I want to tell you that there are three different terms most commonly used in bioinformatics related to this formats, and it consists of, one is known as accession number, another is known as gene ID or GI number, or GI number, another is known as version number. There is a difference between these three. One is known as accession number, other is known as gene ID number or GI number, or other one is known as version number. The NCBI recently means opt out this GI number, and currently accession number and version number are also there. But sometimes you will in the older sequence you will see the GI number also. So what are the difference between these three? For example, I am taking an analogy of a banking system. For example, I went to a bank and I said I want to deposit certain kind of money in your bank. For example, I am saying 1 lakh rupees I want to submit in your bank. Then they will say you have to wait, you will get a token. If I am getting a token, then this token is nothing but a GI number, which is not permanent in nature. But in this case, it is showing me that GI number is a kind of token which is available for me. Second is known as, for example, accession number. When it is when an account, for example, I don't have an account, they will open an account and they will give me unique 16-digit number. So for that, this 16-digit number which remains permanent for me or associated with me, they will do KYC for my account, all the things, detailed information. So this, after approval of this particular thing, for example, I have given my 1 lakh rupees and they check whether my the money bills are, or you can say the rupees or maybe dollars are authentic and they are real, then they assign a real account number to that particular money, that is my account number, or here it is known as accession number, which remain fixed. GI number also remains fixed, but right now it is, we can say it is a temporary thing which is allocated initially when we are submitting certain amount of data in the form of DNA, RNA or proteins. So after that accession number, if some change happen, for example, I have submitted a human insulin sequence, and they have given me a token number, that is a GI number, and after approval that this after checking that this particular sequence is correct, then they allocated the accession number. And after accession number, this accession number is nothing but a alphanumeric number that is given, where one alphabet represent the genus, other represent the species. So this kind of thing is there. So along with that, if certain modification happen, for example, some person X come and he said that all sequences are correct, but there is other information that is also hidden behind that particular sequence, and he is modifying that, or she is modifying that particular sequence, then a version number is allocated to that. For example, my initially accession number is given to me is AB something 0001, and after version number 1 has changed that, then it is 1.1. For example, if I am able to see after a long time that my sequence has accession number has been changed, or it into, for example, AB0001.10, it is not accession number, it is version number, means 10 people have already changed that particular thing. So there is a difference between GI number, accession number, and version number. These all three are associated with each other, and they are interrelated to each other. So in case of Genbank, three things are there. One is known as header, second is known as features, and third one is known as sequence. So second we are talking about, that is features. It consists of detailed information. I will go towards this hands-on session also, so that you will able to identify which things are what. So for example, here you can say features. Here one represent the locus, and other is distinguished, other qualified, that is location, is distinguished by a forward slash. Here source is given. For example, the source length, sequence length is 12496. As you can say, the previous thing we will see. So here the locus number is given. Here the length of the sequence is given. The type of sequence, for example, DNA is there. The linear sequence is there. So for example, sorry, here in this case primates is there, and the date of submission is also there. Along with that, definition is there. Along with that, version number is there. So 2 times modification has been taken place, and there are various keywords that are associated with this particular sequence, that is GC rich region, insulin, polymorphic variation, tandem repeats, all the things are available. And the source of source organism is Homo sapien, and we can see its taxonomical classification also. Along with that, it also gives the reference which authors or authors have worked, or which scientists have worked on this particular sequence, and they have published a this particular paper titled this under the journal Nature. The second reference is this. Other with that, we will see how we can see this particular sequence is available in the database. Second is feature thing. Features thing, you can see organism is there, molecular type is there, and the reference taxonomical number is also there. It also shows the map. Here it is there. For example, in case of chromosome, 2 arms are there, or you can say there is a P and there is Q. So here it is available at 11th chromosome, P8 position, 15.5. So it is there. Where it is, which tissue it is present, liver, and where, but it's a development stage, it is fetus stage. So in case of gene sequence, you already know there are 2 different kind of things that are available. One is known as exon, that is coding part of the DNA. Other one is known as intron. So here 2 exons are there, exon 1 and exon 2. For example, here it is intron 1, and for example intron 2, poly A tail, for example, also there. Or for example, this is untranslated region, this 3 dash, this is 5 dash, all the things are available, or you already know. During splicing, this exon come into picture, and this intron has been removed. Along with that, UTR it is available, which is untranslated region. So in this case of this particular gene, which is starting from 2186 and it is going towards 4969, the whole region is not coding in nature, that is exon in nature. So in this case, it is starting again from 2186, but it is going till 2227. This is known as INS gene or insulin gene. There is certain note that is also added. Number 1, for example, I am saying there are 3 coding regions that are available with this particular insulin gene. So there are various different kind of exons that are also there. Along with that, sorry, for example, I am also saying after this particular thing 2227, the next one is 2228. For example, here it is saying it is available 2186, here it is starting, and here it is 2227. This is the end of that particular exon. After that, this is intron region, which is starting from 2228 and goes towards this particular thing that is known as 2406. So this is the variation, and this is the coding sequence that is available. Here joint term indicates we are joining certain coding region, that is CDAs, whose fragments are ranging from 222, 2424 to 2610 and 3397 to 3542. Here the name again indicates INS, that is insulin, which is a precursor, and protein ID is also there, and you can see methionine is starting. So it's a complete sequence in which it is starting from methionine and ending at N. So we will quickly go towards this hands-on session so that we can see how they look like. So this is the third thing, that is sequence, in which there is origin starting from 1 and ending at certain thing. For example, 4000 something, 900 something, and here double forward slash are there. So we will go towards this particular thing of header and feature thing in our hands-on session. Before going towards this hands-on session, we have to first see what is the meaning of location item. For example, a single number is given, that it means points to a single base of that particular thing is presented, means here it is starting and here it is ending. When two dots are given to it, for example, it is starting at 334 and ending at 889, it means two continuous range of base pairs are included, and the region that lies between start and end consists of that coding region or maybe non-coding region. If certain smaller than or greater than sign is used, here you can see, it means we do not know the exact location, or you can say it indicates the exact lower boundary point of the feature is unknown. We are not sure where it is starting. So in this case, we can say that the endpoint we know 500, but we are not sure where it is starting. So it is, you can say, sometime it is known as a partial sequence when we do not know the complete information. At that point, smaller you can say sign are used. So along with that, here we can say less than 1 means the feature starts before first sequence and continues till 888. For example, here it is, but we are not sure it is not starting from 1, but before that, that is upstream region, it is there. For example, it is greater than sign is there. We can say that first sequence we know it is starting at 1, but we are not sure it is ended at 888. That is, it continues goes beyond that 888. Along with that, if single dot is used, for example 102.110, it means it indicates that exact location is unknown, but that is one of the bases between 102 and 110. That is the region that lies between 102 and 110. We are not sure it is the start point and end point, but somewhere here the coding regions are available, or maybe certain other regions are available. Along with that, if certain this symbol is available, it shows the points of the site which lies between in the basis of 123 and 124. We are not sure whether it is starting from 123 or 124. So for doing that thing, we are using this site. Join command, as I earlier told you, in which different region small fragment join together to form a coding sequence. For example, I'm saying my coding sequence start with 12 and ended at 78, first fragment. Second fragment started at 134 and ended at 202. So here it shows when we join, then it shows the contiguous sequence. The complement, as you already know, in case of DNA, A is complemented with T and G is complemented with C. Here the start base complementary is from 126. For example, this is the start point, is the end point. So in complementary, we are starting this and the base is complementary to 34. For example, here is 34, here is 126. So complementary things are available in the reverse direction. Here it is showing. And then complementary join, when we are using these both things together, that is smaller fragment, contiguous fragment along with the complementary sequence. So these are the things which we have to consider in mind while we are looking at the results. Another one is known as join, complementary. So two different fragments, range of fragments are there. Complementary we have to take, and then we have to join together. Along with that, there is a term J, which points to the base of 100 to 202 inclusive in the entry in the database, which primary accession number of this. So accession number is given to that, having this inclusivity in the database with that particular accession number from 100 to 202. So this whole sequence is given in the form of accession number. Along with that, we are adding a small fragment from 100 to 202. Now join, you can see there is a combination. There is a range from 1 to 100. Then an entry in the form of accession number is given. Then again a fragment is given. Whole three fragments are joined together by using a command that is known as join. So bigger fragments are joined together, a contiguous sequence are joined together by using this particular name, that is Joel. So we will see how we can retrieve our sequence in our hands-on session. So this was a theory. In the next session, I will tell you about the hands-on demonstration about FASTA as well as Genbank. Thank you so much.